means almost double the crops we grow. We're taking steps from developing high-tech farming systems to reducing global food waste. In the meantime, the boom of indoor agriculture is transforming the way we think about the future of food. It's debatable whether indoor farming is economically or environmentally viable, but we can't deny it's part of the solution that continues to evolve. Vertical and container farms are the two major structural forms of indoor farming. As of last year, there were 15 commercial plant factories and 3 commercial container farms in the US, with more under development. Most of these operations are in urban areas, such as Potponics in Atlanta. In Asia, indoor farms are even more prevalent, with 450 commercial plant factories and counting. Particularly, those in Japan and Taiwan are increasing drastically in the last 7 years. Crops suitable for indoor systems are mostly high-value vegetables, such as leafy greens, herbs, and microgreens. We can control various environmental factors to grow plants effectively. Among them, lighting is a critical component that we can manipulate to influence plant growth and development. Light-emitting diodes, or LEDs, are becoming more and more popular these days, not only because they are energy efficient and everlasting, but their spectrum can be tailored or customized for different needs. Here, we define blue, green, red, and far red using 100 nanometer wavebands, and they're abbreviated as B, G, R, and FR. So, why is the spectrum so special? Well, the spectrum of the light source can alter plant growth and morphology. Here, we're looking at lettuce with the same genetics but different growing patterns on the different light sources. The spectrum also influences plant pigmentation, or coloration. At first glance, we see two completely different lettuce plants, one being green and the other being red. What if I tell you these are the exact same plant, only 2 days apart? This lettuce plant was previously grown under mostly red light. Now it has been transferred to an environment with a lot of blue and UV light. Every second in the time-lapse is 2 hours in reality. Within 2 days, it's saturated with anthocyanins that appear red. And this is the power of light on secondary metabolism. Current horticultural lighting seems dichromatic, mostly with blue and red, but there's more to it, like far red. Far red may not directly contribute to photosynthesis, but it regulates photomorphogenesis, like the shade avoidance response. So we wanted to see if far red could be useful and how it would interact with blue and red. We postulated that far red would increase biomass and extension growth, but decrease pigmentation. We used green leaf lettuce Rex, red leaf lettuce Cherokee, and basil Genovese. We sowed seeds and during the first 3 days, grew the plants in the growth chamber at 20 degrees Celsius. We used continuous fluorescent lighting of 100 micromoles per meter squared per second during the first day, and then 180 micromoles from day 1 to 3. On day 3, we transferred the plants to another growth chamber at 22 degrees Celsius. There, we grew the plants on the 6 different LED treatments until day 12 for lettuce or day 16 for basil. At harvest, we quantified plant biomass, morphology, and pigmentation. Now let's take a closer look at the LED treatments. We have two blue to red ratios, 30 to 150 micromoles and 90 to 90 micromoles. Within each ratio, we add 30 micromoles of far red. As for controls, we have 180 micromoles of red or blue alone with 30 micromoles of far red. In all these treatments, the photosynthetic photon flux density was maintained the same, 180 micromoles. We measured their spectral distributions and plant height using a portable spectral radiometer. The peak maxima of blue, red, and far red were 450, 660, and 730 nanometers. So, how did plants adapt with or without far red under these treatments? Here are a few examples. First, let's look at shoot dry weight. Means marked with different letters within each cultivar are different according to the Tukey's HSD test. All data are pulled from 2 replications. For lettuce Rex, additional far red didn't affect shoot biomass at the blue to red ratio of 30 to 150. But when this ratio was 90 to 90, far red increased shoot biomass by about 40%. Lettuce Cherokee showed somewhat similar responses, except here far red increased shoot biomass at both blue to red ratios. However, the percent increase doubles at the higher ratio 90 to 90. This means the effect of far red depends on the blue to red ratio and slightly differs between cultivars. Now that we know far red can promote shoot growth, what about roots? This is an example of Basil genovese. Additional far red at both blue to red ratios produced the greatest root dry weight. For lettuce, however, far red had less or no effect on root development. As for plant morphology, we used leaf length as a primary indicator of extension growth. This is lettuce Rex. There are 2 interesting observations here. First, far red increased leaf length at both blue to red ratios, but the percent increase was 20% if the ratio was 30 to 150, and a higher 60% if 90 to 90. Second, when far red was absent, increasing the amount of blue suppressed leaf expansion. But this effect was reversed when far red was present. A higher blue to red ratio with far red actually increased leaf length. Perhaps the elongation caused by far red prevailed over the suppression caused by blue. Therefore, blue and far red seem to have antagonistic effects on leaf expansion. The other lettuce cultivar, Cherokee, showed almost identical responses. Finally, we examined pigmentation of red leaf lettuce Cherokee using a tristimulus colorimeter. The leaf color was defined in the LAB color space. Since we're interested in how green or red the leaf appears, we present A star. Basically, the greater the A star, the redder the leaf. The lower the A star, the greener the leaf. Please note that all of the values here in this chart are negative, although they could be positive in some other cases. Without far red, a higher fraction of blue produced the reddest leaves. This is not surprising, as blue light is known to promote anthocyanin production. At both blue to red ratios, additional far red decreased the A star, or in other words, reduced red coloration. Although we didn't measure anthocyanin concentrations, we speculate that far red may downregulate anthocyanin accumulation by either inhibiting its synthesis or promoting its degradation. In the near future, we plan to engage in more detailed quantification of pigments, including but not limited to anthocyanins. In conclusion, we can add far red to blue and red to increase leaf expansion, radiation capture, and thus plant growth. The effect of far red sometimes depends on the blue to red ratio, so the 3 wavebands do interact. Also, blue and far red have antagonistic effects on extension growth, or leaf expansion, and pigmentation. So, should far red be used in indoor farms? Which light combination is the best? Well, it all comes down to the specific crop traits desired. Sometimes elongation and weight are desired, and other times compactness and coloration are. Knowing what you want, a spectral regime can then be developed to fit the needs, and this may take advantage of far red. We would like to thank Michigan State University Project Green for funding,